they do not contain true nucleus as well as membrane bound cell organelles. So such group of organisms we can call them as bacteria. So these are microscopic, unicellular and prokaryotic organisms. And these were first discovered by a scientist named as Anton van Leeuwenhoek. A scientist named as A.B. Leeuwenhoek, his complete name is Anton van Leeuwenhoek, in the year 1675. So bacteria were first discovered by Anton van Leeuwenhoek in the year 1675. Actually, he observed these organisms in the stored rainwater. So he collected the stored rainwater, and when he examined under the microscope, he first discovered the organisms which are called as bacteria. But he called those organisms, when he observed under the microscope, he called those organisms as wild animalcules. So he called those organisms as wild animalcules. Later in the year 1838, another scientist who coined the term bacterium. So the term bacterium was given by Ehrenberg in the year 1838. Actually, bacteria were discovered by Anton van Leeuwenhoek, but he called those organisms as wild animalcules. Later, Ehrenberg he coined the term bacterium. And the study of bacteria is called as bacteriology. You may have seen different branches of biology. Likewise, the study of bacteria is called as bacteriology. And you know that bacteria can be found everywhere. They are present in almost all habitats. Even some bacteria are present in and on the surface of other organisms as parasites. So this is some information about bacteria. We see the shapes of bacteria. Actually, depending upon the shapes of the bacteria, there are four types of bacteria, namely coccus bacteria, bacillus bacteria, spirillum bacteria, and vibrio bacteria. So these are the four important types of bacteria which are mainly based on the shape. So based on the shape of the bacteria, there are mainly four types. The first one, it is a coccus. So coccus, in the sense, these are the circular or spherical bacteria. If the bacteria looks like this, so these are the spherical shaped bacteria which can be called as a coccus. Example, streptococcus. So this is one kind of coccus bacteria, which means they are circular or spherical bacteria. So the shapes are very important. So depending upon the shapes only we are giving the names. So coccus, nothing but spherical bacteria. I have given one example, streptococcus. And bacillus, these are rod-shaped bacteria. These are rod-shaped. So they may be looking like this, or they may be appearing like a cigar shape. So likewise, so these are commonly a rod-shaped bacteria. Example, lactobacillus. And spirillum. So these are spiral bacteria. Spiral bacteria, they usually they are spirally coiled like this. They are spirally coiled like this. Such type of bacteria are called as spirillum. Example we can give, there are different species of spirillum. So spirillum is a kind of species, so we can give spirillum as an example. Even spirilla is another example for spirillum bacteria. And vibrio, these are comma-shaped bacteria. So they look like this. Comma-shaped bacteria, example, vibrio cholerae. Vibrio cholerae, it is the example for vibrio bacteria. So try to understand, depending upon the shape, bacteria are classified into four major types: coccus, bacillus, spirillum, and vibrio. So coccus, these are spherical bacteria, example streptococcus. Bacillus, rod-shaped bacteria, example lactobacillus. And spirillum, these are spirally coiled bacteria, example spirillum or spirilla. And vibrio, these are comma-shaped bacteria, example vibrio cholerae. So this is a types of bacteria depending upon the shape. into two types of autotrophic nutrition and heterotrophic nutrition. So here we can see autotrophic and heterotrophic. Autotrophic and heterotrophic. So if they are autotrophs, they can prepare their own food, and heterotrophs usually depending upon other organisms. Even the bacteria may be autotrophic or heterotrophic. Again, under autotrophic, we can see photoautotrophic and chemoautotrophic. So again, under autotrophic mode of nutrition, we can see two different types of autotrophic mode of nutrition. One is photoautotrophic nutrition and other one is chemoautotrophic nutrition. If they are photoautotrophic nutrition, they will synthesize their own food by utilizing sunlight as a major source of energy. So by utilizing sunlight, they will prepare their own food. Such organisms are called as a photoautotrophic. Example, we have studied in the previous class, cyanobacteria. So cyanobacteria, they are photoautotrophic. Then chemoautotrophic. If they are chemoautotrophic, these chemoautotrophic organisms, they usually oxidize or they help in breakdown of certain inorganic compounds, especially nitrates, phosphorus, ammonia. All these inorganic nutrients will be oxidized in order to obtain the ATP, which means, so these organisms will be obtaining nutrition or ATP from the chemicals, like what I said, inorganic nutrients or inorganic substances like nitrates, etc. From them, they will obtain the ATP and they can able to manufacture their own food. And these chemoautotrophic organisms especially helps in recycling of nutrients. So recycling of nutrients in the sense, see, whenever we eat plant products, whatever the organic substances which are present in the plant like protein, carbohydrates, etc., that will be entered into our body. Again, after the death, so our body will undergo decomposition. During decomposition, whatever the inorganic substances will be present in our body, they will be released into the soil. Actually, from the soil, plants will obtain. When we eat plants, that will enter into our body. After our death, again they will enter into the soil. That is called as a recycling process. These chemoautotrophic organisms, they mainly help in recycling of nutrients. And next type, it is heterotrophic mode of nutrition. These are the most abundant type. Most of the bacteria are heterotrophic in nature and they are decomposers. Most of the heterotrophic bacteria are decomposers. So what do you mean by decomposition? Decomposition means the breakdown of complex organic material into simpler inorganic nutrients. I will give you one simple example in order to understand the decomposition process. See, as I said, when we eat plant products, proteins, carbohydrates which are present in the plant that enters into our body. Even in our body, we have proteins, carbohydrates, everything. So after our death, for example, you can take if any plant or animal is dead, so that cannot remain as it is. It will undergo decomposition, which means it will undergo breakdown. That breakdown will be done by various microorganisms, so that the protein is an organic molecule present in that organism, the dead organism. So that protein will undergo breakdown. So proteins will be broken down to form amino acids. Amino acids are further broken down to form ammonium ions. Likewise, whatever the organic materials will be there, that complex organic material, that complex dead body will be decomposed into small inorganic nutrients. That process is called as decomposition. So majority of the heterotrophic bacteria are decomposers because they mainly take part in decomposition process. Again, in heterotrophic mode of nutrition, we can see the saprophytic bacteria, parasitic, and symbiotic bacteria. So under heterotrophic mode of nutrition, there are saprophytic, symbiotic bacteria is rhizobium. Rhizobium. This rhizobium, it is usually present in the roots of the leguminous plants. In certain leguminous plants like as you see pea, groundnut, all these are called as leguminous plants. In their roots, there is one specific bacteria, that is a rhizobium. Then there is a mutual association between the rhizobium bacteria and the legume plants. So if it is a mutual association means both should help to each other. So how the rhizobium will help the plant? This rhizobium actually it helps in nitrogen fixation. It will convert the atmospheric nitrogen into useful forms of nitrogen. That nitrogen will be required by the plant. In the previous classes only I told you the atmospheric nitrogen cannot be directly utilized by the plants. It has to be converted. So that conversion will be done by rhizobium. So rhizobium will convert the atmospheric nitrogen into useful forms of nitrogen. So it will be providing inorganic nutrients to the plant. Then that plant also should help the rhizobium. How the plant will help? So that plant will prepare the food for this rhizobium. Even it provides a shelter for this rhizobium. So this rhizobium obtaining food and shelter in the plant. In turn, it provides inorganic nutrients for the preparation of the food. Likewise, if both are benefiting, not a single organism is benefiting, if both are benefiting, that is called as mutual beneficial association. So this is symbiotic mode of nutrition in which the bacteria will be living in some other organism in a mutual beneficial association. It will obtain food and shelter in the host organism. In turn, it will provide the inorganic nutrients. Best example is rhizobium. So this is about the mode of nutrition. Now we will discuss the ultrastructure of bacteria. It is very important for your examination. Most of the time you can expect this question. Explain the ultrastructure of bacteria. So now we will discuss ultrastructure of bacteria. You know that already bacteria. These are unicellular microscopic organisms with prokaryotic cell structure. Why they are prokaryotic? They do not have a true nucleus or any membrane bound cell organelles. Hence they are prokaryotic. And so the size usually varies from the size of the bacteria varies from 0.2 to 1.5 micrometer in diameter. The diameter usually varies from 0.2 to 1.5 micrometer and length is about 2 to 10 micrometers. So this is the size of the bacteria, diameter and length of the bacteria. And when you observe the diagram, so this is a typical bacteria. So which shape of the bacteria is this? So this is a rod-shaped bacteria. You can say it is a bacillus type of bacteria because bacillus in the cells they are rod-shaped. A typical bacteria what we are going to study, it is a rod-shaped bacteria and which is surrounded by a rigid cell wall. It is very important. This typical rod-shaped bacteria which is surrounded by a rigid cell wall and this cell wall is made up of a chemical component that I told you before only. So cell wall of bacteria is mainly made up of peptidoglycan. So it can also be called as a murine. Peptidoglycan or murine. So rod-shaped bacteria enclosed by a rigid cell wall which is made up of peptidoglycan or murine. So what the layer you can observe in this blue color layer, the blue color layer I am saying it as cell wall. So that is made up of peptidoglycan. Then outside the cell wall, you can observe outside this blue color layer, you can observe a red color layer. So outside the cell wall, you can observe a thick layer called as capsule. The outermost layer, the outside the cell wall, we can call there you can study in daytime. Now just listen. Plasma membrane, it is made up of phospholipids. And this plasma membrane, you can observe the innermost layer is a plasma membrane. The plasma membrane will be showing projections here and there. So all these projections, what you can see, they are arising from the plasma membrane. Now I am talking about the plasma membrane will be folded inwardly. You can observe the plasma membrane which is folded inwardly. These structures are called as mesosome. The inward foldings of the plasma membrane are called mesosomes, and these mesosomes contain oxysomes. So when you observe the structure of the mesosome, you can able to know the oxysomes, and these oxysomes mainly helps in respiration. Actually, in eukaryotic cells, the mitochondria is the site for respiration, and mitochondria is a membrane-bound cell organelle. Here, as we say prokaryotic cell, there are no membrane-bound cell organelles and no mitochondria. The function of mitochondria will be performed by this mesosome. So you should remember this. Mesosomes are equivalent to mitochondria because in eukaryotes, mitochondria helps in respiration, but in prokaryotes, mesosomes helps in respiration because they contain oxysomes. Next, so this is the cytoplasm, the fluid matrix inside the plasma membrane. So the fluid matrix is called as cytoplasm. What the dotted line structures you can observe, this is the fluid matrix which is called as the cytoplasm. And this bacterial cytoplasm, it contains different structures. Mainly it contains nucleoid, which is also called as incipient nucleus or false nucleus or genophore. There are different names given for this nucleus. You just remember it is incipient nucleus or false nucleus. So why it is called as incipient nucleus? Because there is no nuclear membrane around the genetic material. In all eukaryotes, the genetic material will be surrounded by a specific nuclear membrane, but here there is no nuclear membrane. The genetic material is naked. It is freely suspended in the cytoplasm. Such a naked DNA is called as nucleoid or incipient nucleus, and this incipient nucleus contains a double-stranded DNA molecule. So this is the double. You can observe two strands which are coiled like this. So these double-stranded DNA molecule, it is nothing but nuclei. And thus bacterial cytoplasm also contains 70s type of ribosomes. In the previous class, I have asked you how. So 70s ribosomes are made up of two subunits, a smaller subunit and a larger subunit. So smaller subunit having 30 years and larger subunit having 50 years. But how it becomes 70 years? Actually, we are not adding these two subunits when they are subjected to centrifugation. So depending upon the sedimentation coefficient, at what rate these subunits will be undergo sedimentation. So based on that, we will be saying 70 years and 80 years. That you will study in detail in the chapter cell. Now we just remember the bacteria encloses 70 years type of ribosomes. Then what is the main function of ribosomes? Actually, ribosomes mainly helps in protein synthesis. That's why ribosomes are called as protein factories. Ribosomes are called protein factories because they are the main sites for protein synthesis. And even the cytoplasm contains the reserve food material in the form of glycogen. So glycogen, it is the reserve food material. Not only glycogen, they may even contain volutin or oil droplets. They may contain volutin or oil droplets. See, even in our body we have reserve food material, that is glycogen only, and plants have reserve food material in the form of starch. Likewise, there are different types of reserve food materials will be there. So even in bacteria, the reserve food material will be stored in the form of glycogen, volutin or oil droplets. Then here you can observe one more structure called as plasmid. self-replicating, extra chromosomal, double-stranded, circular DNA. You don't get confused with it. Just remember, these are the circular double-stranded DNA. They are called as plasmid. Then what is the difference between this DNA and this DNA? Actually, this is the main genetic material which will be transferred during reproduction. But this plasmid, it is extensively used in the field of biotechnology, and even this plasmid also provides resistance to this bacteria that you will study later. You just remember, it is also very important. You must have to draw in this bacterial cell. So it also contains, the cytoplasm also contains a circular double-stranded DNA called as plasmid. And you can observe from the plasma membrane, in some bacteria, from plasma membrane, a structure will be arising. It is called as a flagella. So flagella arising from the plasma membrane, and it helps in locomotion or swimming. If bacteria is moving, with the help of flagella it is moving, and flagella acts as a locomotory organ in case of bacteria. So this is the long tubular structure called as flagella, helps in locomotion. And here and there you can observe some tubular filamentous structures arising from the plasma membrane. You can observe the difference. So this is a long tubular structure called as flagella, but these are a smaller tubular filamentous structures arising from the plasma membrane here and there. These are called as sex pili, and sex pili mainly helps in the reproduction or conjugation. Conjugation. I told you, when another bacteria comes near to this bacteria, through this sex pili, conjugation tube will be formed. The genetic material from this bacteria can be transferred to another bacteria. That is called as conjugation process. So sex pili, they mainly helps in sexual reproduction, that is conjugation. So this is about the structure of bacteria. All membranes, all structures are very important. Cell wall made up of peptidoglycan, capsule outermost layer made up of polysaccharides, plasma membrane is made up of phospholipids. Plasma membrane shows inward proteins called as mesosomes, and they mainly helps in respiration. Then bacterial cytoplasm contains nucleoid made up of double-stranded DNA molecule, and ribosomes, which are 70S type, mainly helps in protein synthesis. The reserved material in the form of glycogen, volutin, oil droplets, and also contains a circular double-stranded DNA called as plasmid, and the locomotory organ is called as flagella. So the tubular filamentous structure is called as sex pili, mainly helps in sexual reproduction. Actually, the bacteria mainly reproduces by binary fission. You may be knowing about this binary fission. Actually, this bacterial cell, one bacteria divides to form two bacteria. Again, they will undergo binary fission one more time. Likewise, many number of bacteria can be formed. That is called as a binary fission. This bacteria mainly reproduces by binary fission, and during unfavorable condition, it produces spores. Unfavorable conditions in the cells. For example, if this bacteria can survive in water, that is favorable condition. If there is no water for the bacteria, that is called as unfavorable condition. So during unfavorable condition, bacteria produces spores, and rarely it undergoes sexual reproduction either by conjugation, transformation, and transduction. You know conjugation, so the conjugation tube will be formed, the genetic material will be transformed to one bacteria to another bacteria. And transformation, so transformation in the sense, the genetic material will be taken out and introduced into another bacteria. Not through this conjugation tube, this genetic material will be taken and introduced into another bacteria. That is called as a transformation. And a transduction. So in the transduction, a virus particle is involved in order to take this genetic material and it will be injected to another bacteria. For example, bacteriophage. You will study in this chapter only. The bacteriophage, it is a virus particle which will be attached to this bacteria. It will take the genetic material and it will be injected to another bacteria, where this genetic material will be transferred to another bacteria. That process is called as a should note down all the points for this bacteria. And lastly we need to study about mycoplasmas. So lastly we are studying mycoplasma. So under kingdom Monera, the mycoplasmas are the last type of organisms, and these are the organisms without cell wall. They completely lack cell wall. You cannot find any cell wall like that. So the very characteristic feature of mycoplasma is they do not contain cell wall. So they lack cell wall. And the mycoplasmas are the smallest living organisms. They can survive without oxygen. This is also another important point. So they are the smallest living cells because the size varies from 0.3 to 0.5 micrometer. These are the smallest living organisms which can survive without oxygen. That is very, very important. They do not contain cell wall and they do not require oxygen. That's why they can survive in the absence of oxygen. And these mycoplasmas are also called as PPLO. PPLO is nothing but pneumonia-like organisms. Pneumonia-like organisms. PPLO. The mycoplasmas are also called as PPLO, which means pneumonia-like organisms. Why they are called as pneumonia-like organisms? Because they cause a specific disease in animals, which is called as pneumonia. Hence, they are also called as PPLO. And many of the mycoplasmas, they may be saprophytic or parasitic, but most of them are pathogenic, and they usually cause diseases in plants and animals. As I specified, it causes a specific disease in animals called as pneumonia. And another interesting thing, because of the absence of cell wall, as they do not contain cell wall, they can able to modify their shape. We cannot say only this type of shape can be seen in mycoplasma. They are pleomorphic. We will say pleomorphic, which means their shapes will be existing in different shapes because the absence of cell wall. So that is about mycoplasma. Mycoplasmas, they are completely lack cell wall and they are the smallest living cells. They can survive without oxygen, and they are also called as PPLO, pneumonia-like organisms, and many of them are pathogenic. So this completes mycoplasma, and in this class this completes kingdom Monera. So I will conclude the class. In the kingdom Monera we have discussed the salient features, the classification, that is Archaebacteria and Eubacteria. Under Eubacteria we have studied cyanobacteria, bacteria and mycoplasma. This completes the kingdom Monera. In the next class we will start up with the next kingdom, that is kingdom Protista, and we will discuss few objective questions related to this class. See the first question, heterotrophic bacteria helps in. So heterotrophic bacteria I have discussed. So the mainly helps in. See option A, curdling of milk. Option B, production of antibodies. Option C, nitrogen fixation in leguminous plants. Option D, all of these. Actually, heterotrophic bacteria helps in all of these. I told you they are the most abundant organisms. Curdling of milk. See, during curdling process, you will add a small amount of curd to the milk. Later, the complete milk gets converted into curd. So at that time, the lactobacillus will be taking place. That is also a heterotrophic bacteria because it will be obtaining the nutrition from the chemicals, not from the sunlight. Even production of antibiotics. For example, penicillin. You may be knowing that penicillin is most important antibiotic. Not only that, and penicillin, even different types of citric acid, organic acid, all these things will be produced with the help of the microorganisms. They are heterotrophic. And nitrogen fixation in leguminous plants. Yeah, during nitrogen fixation, the rhizobium will be taking place. So there is also heterotrophic bacteria. So option D is correct. All of these. Next question 2. Select the total number of diseases from the following caused by bacteria. So here they have given some examples of diseases. You need to find out how many diseases will be caused by bacteria. So they have given mumps, smallpox, cholera, typhoid, tetanus, sleeping sickness, and malaria. So these are the diseases. And options they have given: option A 2, option B 4, option C 5, option D 6. Here option B is correct, 4. So out of these diseases, 4 diseases will be caused by bacteria. I will tell you. So firstly they have given mumps. Actually mumps is caused by a virus, and smallpox is also caused by virus. Citrus canker, it is caused by bacteria Xanthomonas. First one, cholera, it is caused by Vibrio cholerae, it is also correct. Typhoid, it is also caused by bacterium. Even tetanus is also caused by bacterium. But sleeping sickness is caused by protozoa. Even malaria is also caused by protozoa. So only 4 diseases among these will be caused by bacteria. So for this class it is enough.